Right. Good morning, good afternoon, and good evening, everyone. My name is Asli Heitzer, and I'm a senior project manager on HIV prevention research and development at the Global HIV Vaccine Enterprise of the International AIDS Society. And it is my great pleasure today to open this timely topics webinar on cutting-edge developments in HIV vaccine research, new insights and implications. And I would like to welcome all participants as well as our speakers and our moderator. Our webinar today aims to provide an overview of recent advances addressing the challenges of inducing broadly neutralizing antibodies, the development of germline targeting epitope scaffolds, and the use of innovative vaccination regimens. We also would like to raise awareness of ongoing HIV vaccine R&D, highlighting the continuous commitment of the scientific committee, it's not forgotten, to developing an HIV vaccine as another prevention tool. So today's meeting is comprised of 6 presentations, and each of these presentations is followed by a 7-minute Q&A. I truly encourage you to use the Q&A function and ask your questions. And please note that this meeting is being recorded, and by participating, you also consent that the recording will be used publicly for educational purposes. I would now like to introduce our moderator and presenter for this webinar, William Schief. William Schief is Vice President, Antigen Design and Selection in Infectious Disease Research at Moderna, Professor for Immunology and Microbiology at Scripps Research, and Executive Director, Vaccine Design at the IAVI Neutralizing Antibody Center at Scripps Research. And he works on a wide array of vaccine and biotherapeutic targets at Moderna, and at Scripps and IAVI, he focuses on HIV vaccine development using the germline targeting strategy in collaboration with Moderna and other partners. And the work focuses on computation-guided and structure-based design of immunogens and immunization regimens with the goal of inducing broadly neutralizing antibodies against HIV, but also other pathogens that have frustrated traditional vaccine design strategies. And with that, Bill, please, the floor is yours.
Thanks, Asli. It's great to be here. And I see Nepal, Liberia, Tanzania, Ethiopia, Chile, Haiti. So it's great to be able to present in front of all of you, and we'd love to get your questions. Today, we're going to give several presentations showing preclinical data that we think is making really good progress toward HIV vaccine development. And I'll start off with an overview. So let me share my slides. This is a model of the HIV glycoprotein spike, very similar in many ways to the SARS-CoV spike. The blue structures are flickering glycans that are put on by the host cells that are infected. The red surface is surface that's highly variable from one HIV strain to another. Sorry, it stopped. And the yellow surface is a surface that's less than 80% conserved. So just like making a COVID vaccine, we want to induce antibodies against the spike to prevent HIV envelope infection of target cells. But because it's so variable and so highly glycosylated, it's very difficult for antibodies to grab on in a manner that'll allow them to neutralize anything more than one particular strain. And so the structure, glycosylation, and sequence variation pose major challenges to the development of broadly neutralizing antibodies, which is what we need. Fortunately, through the work of many people in the field, HIV broadly neutralizing antibodies, or bNAbs, have been discovered. They neutralize diverse isolates. They can protect against infection in non-human primates and in humans. Different classes of bNAbs bind to different epitopes on the HIV envelope. Here's a model of the envelope with 4 different classes of bNAbs bound. And we believe that an HIV vaccine based on bNAb induction will likely need to induce multiple classes of bNAbs in order to achieve sufficient neutralization, potency, and breadth to protect against diverse isolates circulating among humans. And we, in the work that we're doing in the Scripps CHAV-D at the IAVI Neutralizing Antibody Center in collaboration with Moderna, in collaboration with many collaborators, we're aiming to induce broadly neutralizing antibodies against 4 epitopes for complementary neutralization coverage in terms of titers and breadth. And the four are shown here: the CD4 binding site, VRCA1 class, the V2 apex, PCT64 and PG9, the V3 glycan epitope, BG18 and PGT130, and EMPER, where we're going for 10E8 and related antibodies. And just in the last few months, we've published a bunch of papers related to three of these epitopes. And today we're going to have talks from Chris Cottrell and Xuansong Wang about the CD4 binding site, John Steichen and Zhenfei Zi about the BG18 targeting and V3 glycan, and Torben Schiffner about 10E8 antibody going after the 10E8 in induction for EMPER. And so they're going to be talking about this strategy that we're pursuing. And let me just give you two slides about that. So what is required to induce protective BNAbs? You know, no matter what strategy you pursue, this is sort of what we think needs to happen. The vaccine needs to engage naive B cells, of which most of them will be garden variety blue naive B cells that don't have a chance of becoming a broadly neutralizing antibody, and a very small minority, rare naive B cells like this red one will have the right genetic properties that will allow them to develop with somatic hypermutation into a BNAb. These are rare. They have specific genetic features for each class of BNAb. For each class, there's a diverse pool of them that have the right specific genetic features, but are otherwise very diverse. And they're difficult to activate with HIV wild type proteins. So developing an immunogen that can turn those on is very difficult, but we need to do that. And through vaccination, after triggering the right naive B cells, vaccination needs to increase somatic hypermutation in those B cells and switch them to becoming plasma cells to secrete BNAbs. So whatever strategy you pursue essentially needs to accomplish these goals. And the strategy that we and our colleagues are pursuing, we refer to as germline targeting vaccine design. And that's because it really keys off the priming immunogen, which we refer to as a germline targeting prime. That's an immunogen that's designed specifically to find those rare naive B cells that have genetic features necessary to develop into a BNAb. And the priming immunogen has to be able to target a broad pool of those. They're still rare, has to be able to target a broad pool of them that are diverse, but still share the necessary genetic features. And that germline targeting priming immunogen is supposed to trigger the naive B cells, get them to expand, get them to create a pool of germinal center and memory B cells that are slightly more mutated in the right direction. They have a little bit of somatic hypermutation. And then our plan is to give a little bit more native-like boost immunogen, the first boost, and that needs to trigger that pool of GC and memory B cells and kick it further in the right direction. And in the iterative vaccine development process, we're trying to figure out how many heterologous boosts are required to eventually produce BNAbs in memory and then kick them to plasma cells. But overall, we refer to this process as germline targeting vaccine design. And the talks today are going to talk to you about early stages, the priming and the first boosting stages of this process. We did do a clinical trial with a whole bunch of collaborators, many co-authors, and we published this in 2022 that showed, for one example, for the case of VRCA1 BNAbs, we could use a nanoparticle priming immunogen called EOD-GT860-MER to trigger the right naive B cells and induce VRCA1 class responses in 97% of the participants. And the only one participant that didn't make a VRCA1 class response was genetically incapable of doing so. And that's one reason why we're trying to induce multiple different classes of BNAbs so that no vaccine recipient is left out, if you will, that they're always able to make one or two different kinds of BNAbs, even if they can't make all four. So we do believe this was a major step ahead for the field to show that this concept of priming actually practically can be done in humans. And if you haven't seen the paper, you should check it out. Not only did it induce these precursors in 97% of the participants, but the frequencies in each person's, in their PBMCs in their blood were very high. It made us feel like, okay, the pool of VRCA1 class B cells that it induced is big enough to be susceptible to boosting by the next immunogen. It seemed very promising. And so what are the next major milestones? So that trial has established proof of principle for germline targeting vaccine priming in humans. we think the next major milestones at a very high level are we need to demonstrate heterologous boosting toward BNAB in humans. If this whole sequence of heterologous immunization is going to work, we need to show at least once that one heterologous boost does advance the immune responses in humans. That hasn't been done yet, and that was tested in a clinical trial, AVI-G002, and that manuscript is under review. We're not going to talk about it today, but we've talked about it in other forums. The next more advanced step would be to literally induce broadly neutralizing antibodies of one particular class in humans. We get through this whole sequence and show that we can induce BNAbs for just one class. And then we'd like to move really much closer to what a vaccine needs to look like and induce BNAbs in humans for multiple classes in parallel. And I have a note down here that we believe it's really important to establish these milestones in Africa, where an HIV vaccine is most needed, where the burden is highest. And IAVI-G003 did test priming in Rwanda and South Africa, and that manuscript is also under review. And Eunice Ndwati is going to give a talk about that at R4P next week. So today, in today's webinar, we're going to be focusing on preclinical data, and two different talks from Chris Cottrell and Shusong Wang will give you preclinical data that provided support for G002 priming and boosting, and G003, like I mentioned. John Steichen and Zhenfei Zi will talk about preclinical data for BG18 priming and also prime boost. And the priming is being tested in a clinical trial HVTN 144 currently. And then Torben will talk about a really challenging problem of priming for the 10E8 Emperor class. And to accelerate vaccine development and enable us to do all of the phase 1 clinical trials that we believe will be needed to iteratively learn how to develop this sequence of immunogens to induce broadly neutralizing antibodies in multiple classes of broadly neutralizing antibodies, we're partnering with Moderna to accelerate that development and clinical testing and to enable delivery of membrane-bound trimers, which in our studies with Facundo's lab and others, we've shown that there's some advantages, and also in the clinical trial HVTN 302 with Julie McElrath in the HVTN, we've shown that there's advantages to membrane-anchored trimers, which would be very difficult to deliver by protein. And so I just want to thank, to do all this work and everything that you're going to see requires collaboration and funding and partnership of many different institutes and many different funding agencies, obviously. And I mentioned G001, just wanted to thank the study volunteers and the many people involved in that study. And I'll stop there and I will stop sharing. And we're going to have Chris Cottrell come on and talk about preclinical data that led to G002. And then there's going to be questions after he talks, I believe. So, Chris, if you want to share your slides.
There you go. Yeah, so I'm going to talk about heterologous prime boosting with mRNA immunizations in humanized mice, targeting the VRC01 class epitope, CD4 binding site epitope. So we define VRC01 class BNAbs as VH12, STAR02, or STAR04 heavy chain alleles paired with light chains that contain 5 amino acid CDRL3s. And there are numerous examples of VRC01 class BNAbs that have been elicited from people living with HIV, and they use a variety of different light chains, both kappa light chains and lambda light chains. The VKappa 133 subclass of VRC01 class BNAbs is particularly interesting to us because they don't require deletions in the CDRL1 in order to accommodate the N276 glycan. And we believe deletions are difficult to induce reproducibly by vaccination. So the VKappa 133 subclass is an important target for us. And also just to highlight, N6, one of the most highly broad and potent VRC01 class BNAbs, also uses the VKappa 133 light chain. So VRC01 class BNAbs make contact primarily using their CDRH2. So CDRH2 makes contact with HIV envelope in the CD4 binding site. The CDRH2 contacts are shown in brown here, and also with its framework 3 regions was shown in green. And it doesn't make a whole lot of contact using its CDRH3, which is beneficial for germline targeting because we don't rely on specific VDJ recombinations or specific D genes. All we really need is the V gene paired with a 5 amino acid CDRL3 light chain. As Bill mentioned, we developed a multimeric germline targeting immunogen called EOD-GT8. It has 60 copies of the EOD engineered outer domain antigen on a limousine synthase nanoparticle. The CD4 binding site is shown here in yellow. The scaffold, the EOD scaffold is shown in green, and glycans are shown in blue. It's oriented such that the CD4 binding site is actually at the top on the surface of the nanoparticle, and this is showing how VRC01 would bind to EOD and how the scaffold is oriented relative to the limousine synthase. As Bill mentioned, G001 was a human clinical trial testing EOD-GT8 protein in humans. It elicited VRC1 class, activated VRC1 class BNAbs, BNAb precursors in 97% of participants. When we looked at the subclasses, VKappa133 was the most common subclass elicited in G001, further emphasizing its importance for us. So Fred Alt and Silo at Boston Children's developed a knock-in mouse model for us, where one of the V genes in the mouse locus has been replaced with the VH12-STAR02, and the light chain has been replaced with the VKappa133. In this mouse, it undergoes normal VDJ recombination, so we get diverse CDRH3s, both in length and in sequence. And they included a human TDT as well in order to increase the frequency of light chains with 5 amino acid CDRL3s. So when we look at the overall frequency in this mouse, we see that the VRC01 class frequency with VKappa133 light chains is only about 3 times higher than it is in humans, comparing to previously published data. So we think that the precursor frequency in this mouse is at the right level for us to be able to test our immunizations. We also looked at the affinity of these antibodies, and the affinity of these antibodies for EOD-GT8 is nearly identical between humans and between the antibodies isolated from mice. So now that we have a good mouse model, we needed a booster immunogen for boosting responses that were primed with EOD-GT8. We developed a GP120 core immunogen, shown here as the CD4 binding site in yellow. We knocked out the N276 glycan with 2 mutations, shown here in orange, and we added additional engineered glycans to the surface to mask potential off-target responses, shown in blue here. This core immunogen was also arrayed on limousine synthase as a 6-dimer nanoparticle. It self-assembles, and both immunogens are going to be delivered using mRNA LNPs produced by Moderna. So this is our immunization scheme. We prime with EOD-GT8 at week 0. We boost with core T28V2, the booster immunogen at week 6, and then we sack at week 12. We do antigen-specific cell sorting, 10x genomics paired VDGA sequencing, and then we process using a custom bioinformatics pipeline. When we look at the frequency of VRC1 class B cells among total memory B cells, we see that after priming, it's a little less than 1%. When we give the core T28V2 boost immunogen, it goes up to about 1.5%. Which is statistically significant from increase relative to priming. If we give a placebo, there is no boosting effect. And then if we just give core T28V2 as a priming immunogen, we see no VRC01 class memory B cells. If we then look at the frequency of VRC01 class using the VKappa133 light chain, we see that the frequency increases after priming to about 1 in 1200. The frequency at the naive time point was 1 in 600,000, so there's a significant increase in the frequency after priming, and that frequency increases again when we give a boost to about 1 in 200. We also wanted to take a look at the affinity. So these antigen-specific B cells were used to produce recombinant IgGs, and we tested their affinity for EOD-GT8. So here we're looking at the naive precursors from the mice that were isolated with the EOD-GT8, and they have a median affinity in the micromolar range. After priming with EOD-GT8, we see a significant increase in affinity. These antibodies have some affinity for our core booster immunogen, although it's again in the micromolar range. Once we boost with core G28V2, those antibodies again develop a significant affinity for the booster immunogen down into the nanomolar range. And these antibodies also have affinity for the booster immunogen when we have the N276 glycan present. We also wanted to look at the affinity of these antibodies for potential boost 2 candidates. So here we're looking at HIV envelope trimers, either wild type or lacking the N276 glycan, using a variety of different knockout mutations for the N276 glycan. What we see is decent binding to 3 different HIV envelopes: 1428, 1HT2, and 191084. And these could serve as potential booster immunogens delivered as membrane-bound trimers, as Bill mentioned in the introduction. We also took a look at the neutralization of these antibodies, and not surprisingly, they don't neutralize wild-type viruses. However, they are able to neutralize, to some degree, viruses that are lacking the N276 glycan, although it's sporadic neutralization, with some antibodies showing cross-neutralization of heterologous strains lacking the N276 glycan. So just just for a quick summary, the main issues are: can we do germline targeting immunogens activate naive precursor B cells, and can booster immunogens shepherd those B cells to gain favorable mutations? And I showed you that we can not only activate those B cells, but those B cells gain affinity for more native-like booster immunogens. We were able to elicit VRC1 class B cell responses in our naive mice, or our knockin mouse model. And those responses were boostable. And this is showing the first 2 steps of a vaccination series for eliciting VRC1 class B NAbs. I'd like to thank everyone involved in the study, particularly Silo for developing the mouse model, and JH and her team at IAVI for doing the cell sorting and sequencing, and of course funding. Happy to take questions.
Chris, we have one question from Daisy Uya, if I'm pronouncing her name correctly, and I'll read it off because I think you can answer it. Intrigued by the participant who was genetically incapable of producing BNAbs, how did they differ from the others? And does this mean vaccines will need to be tailored according to people's genetic makeup?
Yeah. So that's an excellent question. So the participant that wasn't able to make a VRC1 class response in G001 clinical trial had the STAR05 and the STAR06 alleles for VH1/2, and those alleles are not permissive to making VRC1 class antibodies. And we think that that's roughly the percentage that you would see in the population. About 95% of the population have a permissive allele for VRC1 class, so there'd be about 5% that would not be able to make this type of response. That's why it's important to have other responses elicited in parallel for an effective vaccine.
And I could just add that whenever you're targeting a broadly neutralizing antibody, you're trying to induce that in vaccine recipients, the B cells have a certain set of minimum genetic requirements that allow their BCRs to develop into that broadly neutralizing antibody and to bind HIV envelope in the way that BNAb does. And there's always specific alleles that are needed. And so any one individual may not be able to make one kind of BNAb or another kind of BNAb, but if we're targeting multiple, we're hoping that we can get them through. Okay, Chris, we have another question from Roger Tatude, who's one of the sponsors of this webinar. How far in time are we from a trial in human?
So the immunizations that I showed you have already been tested in humans. The prime and the first booster immunogen, that manuscript is under review now. We'd be looking to test the next 2 immunogens in the series over the next couple of years.
And everyone's asking about human clinical trials, even though you're talking about mice, but that's okay. There's another question that says, how long will it take to have an effective vaccine if things continue to go well? That's from Matt Muchler. He says, what should we tell people when they ask, do we have a vaccine?
I think an effective vaccine is still a ways off. So we're still in the phase 1 study for clinical trials. So obviously, we would still need to go through a significant amount of development and then eventually a phase 3 efficacy trial. So still a ways off.
Yeah, I mean, I think the thing that we are currently aiming for the milestones that I outlined in my talk. We need to show that heterologous boosting works in humans, like Chris showed it worked in this mouse model, and Xu Song is going to show in a different mouse model what happened. We need to be able to induce broadly neutralizing antibodies in a phase 1 clinical trial as a major milestone. And then we'd like to be able to induce multiple broadly neutralizing antibodies in a phase 1 clinical trial. And once we can do that, we'll hopefully be holding on to something that's worth taking forward to phase 2 and phase 3, efficacy studies. But we're probably something like 10 years from that, between 5 and 10 years from that, I think. Okay, I think we should move to the next talk. And so the next speaker will be Xusong Wang from the Facundo Batista's lab at the Reagan Institute, also a member of our Scripps CHOV-D and a very strong collaborator in all the work we're doing. And so I'll let her take it away.
Okay, can you see my slides, everyone?
Yes.
Okay, nice. Currently I'm a research scientist in Batista Lab. So my work is mainly focused on the validation of the HIV vaccine candidates in humanized mouse models. And this work was collaborated with the Shift Lab. So as Bill mentioned that the broadly neutralizing antibodies are really important for protecting people from infection. And so far, there are tremendous of the BNAbs isolated from the patients. So they target the different epitope of the virus bag, such as the Apex, Hymenopatch, CD4 binding site, and PER, et cetera, et cetera. So each person of our lab focus on the different groups. For me, I'm really focused on the VRC01 group, which targets the CD4 binding sites. So since the broadly neutralizing antibodies precise a lot of the somatic head mutations for HIV, almost around the 30% of the somatic head mutation. So the ideal HIV vaccine design is based on the concept of the sequential immunization. Basically, you will design a germline targeting immunogen and followed by immunizing with a series of the heterologous boosters, and to trigger those mutate naive B cells to acquire more and more somatic head mutation and affinity maturations, finally to achieve the mature BNAbs. So previously, the Shinkrati lab identified and isolated a series of the VRC01 B cell precursors, and then we generated the humanized mouse models bearing each of the precursor lineage, and we used the EOD-GT8 6-mer protein for immunization to test the mouse models function. So we could see after priming the B cell precursors can be activated and undergo the somatic head mutation as well as the memory B cell differentiation. So that means that this EOD-GT8 6-mer protein can work very well in the humanized mouse models. And this work was published in the ENDO papers. So next, we isolate each of the B cell precursors then to transfer them simultaneously in the same host and generate humanized mouse models bearing the BCR repertoire. So the goal is to test a new mRNA-LNP formulation encoding the EOD-GT8 6-mer. So what we see that after priming the multiple B-cell precursors can be activated, and this B-cell response can sustain until day 36. So when we isolate the sequence to see the sequence proportion after immunization, we could see the diverse sequence occurred, and the sequence can sustain until the day 36. And then we isolate the monoclonal antibodies to test affinity to both the priming ligand and as well as a series of the booster candidates. So we could see some of the monoclonal antibodies not only acquire the affinity to the priming ligand, but also to the booster ligand. So that means those boosters are potential to target the antibodies, the post-priming. The question is that can this booster induce the response further in vivo and to trigger the B cell to acquire more somatic head mutation and affinity maturation. So to answer these questions, we immunize these humanized mouse models with a prime booster strategy. So here we screen the G5 and G28 booster candidates, and we do see after prime booster strategy, so they generate the much higher B cell response compared to the prime only group. And after we isolate those sequence and we test the monoclonal antibody affinity as Chris showed the method, so we do see the boosters can triggers those B cell to acquire more somatic head mutation and undergoes the affinity maturations. So next, we cooperated with Chris, and Chris modified the G28 boosters and generate a new version called the G28 version 2. So we compare those two booster candidates in vivo to see how it looks for inducing the B cell response. Here we see the G28 version 2 induce the slightly better B cell response compared to the G28, and here is the kinetic. The response kinetic over time. And when we isolate those sequence and test the affinity maturation, as we previously mentioned, so we also see there's the new version G20 version 2 trigger this B cell acquire more somatic head mutation and go the affinity maturation further. And so the next step is to collaborate with our co-workers to design more native-like immunogens. So hopefully we can achieve the BNAP after these sequential immunizations. So to summarize these slides, though, we use the humanized mouse models bearing the many B cell repertoires to test a series of prime boost mRNA-LNP vaccine candidates. And after priming, we could see multiple B cell precursors were activated and generate some somatic head mutations. And after boosting, we could see they acquire more somatic head mutations and undergoes the affinity maturations toward the mature like BNAP. So in our mouse models, we validate the EOD-GT8 6-mer and a modified booster. And now, as Bill said, that those vaccine candidates are undergoing the clinical investigation. So, and our preclinical mouse models were published in the Science Immunology paper. So I will thank our lab members from both the Batiste lab and Shift lab, and I will thank the Moderna for the mRNA-LNP formulations. I will thank the fundings from the NIH and Bill Gates Foundation, and also I will thank the organizers. Thank you very much for providing me this good opportunity.
Great. Thank you, Xuansong, for a fantastic talk. And we're hoping for more questions.
Mm-hmm.
And I see some coming in. Well, so there's one question about, okay, so here's a question from Roger. And he says, you generated a new version of the best booster, but would you consider mixing the boosters?
So I think the target is like a similar aptope for those booster candidates. So I think if you want to make the heterologous booster, so the ideal goal is to induce like a diverse induce the mob-wise the diverse response rate. So that is better to use the heterologous booster to target like a different aptope or the different stage of those sequence. But what I can say that those boosters I tested, like G28 or G28 version 2, is like they target the similar aptope. So maybe Chris can explain more about those boosters, right?
I don't know.
Yeah,
Chris, go ahead. Yeah.
the overall frequency just increased with Core D28V2. That's our boost number one immunogen. As far as mixing them with the other ones, there's not much of a gain there. It's just a difference in the resurfacing and the glycan presentation on the different booster immunogens. So we don't think mixing them would drive additional SMAC mutation.
Okay, there's another question that says, how will you select the country where clinical trials phase 1 will take place? And I can just answer that by saying there's many different institutions and individuals involved in planning the clinical studies across HIV vaccine research. For the clinical trials related to the work we're talking about here today, our sort of germline targeting research, so far, we've done clinical studies in the United States at many different sites in the United States and in Rwanda and South Africa. And one of the constraints that we face with germline targeting is to analyze the response and to figure out if the vaccine elicited the responses that were intended. We need to do very, very detailed and sort of expensive and highly technical analyses. Involving B cell sorting and B cell receptor sequencing and bioinformatic analysis. And we've got that set up in several different labs in the United States, and actually now in a lab in Kenya, where Eunice Ndwati's lab analyzed the responses from IAVI-G003, and in some labs in South Africa. And it's probably that kind of analysis is probably available elsewhere, but in our sort of collaborative network, we haven't made those connections yet. I do think over time, the countries may expand, but there are lots of considerations. I think I should also point out that so far, at least for IAVI-G001, where we had very positive clinical data, we had similar experiments in different mouse models to those described by Chris and Xuansong here. And the results in the mouse models were fairly predictive of what happened in humans. Maybe not to the highest level of detail, but overall, they were quite predictive. And that's very helpful for our field, because that means that, we can do lots of iterations in mouse models before we go to humans, so that when we do go to humans, we have a pretty high, at least we can be, we believe there's a high chance of success that we're going to get the readout that we expect in humans based on our preclinical data. So, the studies that Chris and Xuansong described, we think are super important.
Thank you, Bill.
Yeah. Well, so why don't we keep on going? Let's move to the next speaker, and that's going to be John Steichen from Scripps, and he's going to pivot to a different bNAb target. He's gonna go to a bNAb that's trying to bind to the V3 glycan epitope, and it presents unique challenges, and it kind of gives a different perspective on how complicated germline targeting is. So take it away, John.
Okay, thanks, Bill. Okay, yep. So I'm going to be talking about a new epitope, the V3 glycan, and I'm going to be talking about vaccine priming of rare HIV broadly neutralizing antibody precursors. And I'm mostly going to be focusing on these two papers we published in Science. The first one in 2019, we basically described a new method for germline targeting that we call HCDR3 dependent germline targeting. That stands for heavy chain complementarity determining region 3 of the antibody. And so we described a method in 2019, and then this year we published a paper in Science where we basically validated that method in rhesus macaques. And so I'm mainly going to be focusing on this newer paper. And so the antibody we're targeting is called BG18. It's a HIV broadly neutralizing antibody that, as Bill mentioned, binds to this V3 glycan epitope. And so that's shown here. So this in gray is the HIV envelope protein that's on the surface of the virus. And it basically covers itself in sugar molecules, and that shown in green here, and that allows it to hide from the immune system. And the way BG18 can neutralize the virus is it has this long loop here shown in blue. This is called the HCDR3 loop. And you can see it reaches past the sugar molecules. It reaches down through the sugars to contact the gray surface underneath. And that's how it's able to recognize the virus. And so this antibody can neutralize about 63% of global HIV isolates. And we believe the precursor antibodies that can mature into BG18 class antibodies are present in most people. We think most people should be able to make a BG18 class response. That's why we think it's a reasonably good vaccine target. The one challenge of this antibody is that the B cells that encode BG18 occur at a very low frequency in humans, which is in part due to the reliance on this HCDR3 loop, which is rare. But we think it's important to target this type of antibody because most antibodies do rely on their HCDR3 loop for binding. So this is the normal mode of binding for an antibody. So we think this is a more generalized way of doing germline targeting. The previous two talks on VRC01, that is a special case, that antibody that doesn't rely on the CDRH3, but we would like to be able to target most types of antibodies. So this is an important method to work out. And so we developed an immunogen called N332GT5. So it's based on an HIV envelope protein, BG505 SOSIP. And we use the strategy of HCDR3 dependent germline targeting to design it to bind to BG18 class antibody precursors. And when we're doing vaccine studies, the way we know if we've activated a BG18 class B cell. The way we know a B cell is BG18 class is we can sequence the antibody gene in the B cell, and we look at the sequence of that HCDR3 loop on the antibody. And if it has a specific sequence motif in that loop, then we call that a BG18 class antibody. And so one challenge of germline targeting is that we're specifically targeting human antibody genes. And so not every animal, animals have different antibody genes. They're not all exactly like the human versions. And so it can be difficult to find an appropriate animal model. And so we looked in rhesus macaques for BG18 class B cells, and we found that the majority of them do have BG18 B cells with that HCDR3 sequence motif. And so each of these circles represents the frequency of BG18 in one rhesus macaque. So we looked at 60 of them. The majority have it. compared to humans, they're a little bit more frequent in humans. So we think rhesus macaques are a little bit more stringent of an animal model to test BG18 vaccine design. And so we carried out an experiment, a priming experiment we used 8 rhesus macaques to test this N332 GT5 priming immunogen. We used SMNP was the adjuvant, which was developed by Daryl Irvine's lab. It's a strong adjuvant. And we delivered the immunogen by an escalating dose a priming protocol where we give an exponentially increasing amount of the priming immunogen over the course of 2 weeks. And then at week 10, we give a bolus single shot of the immunogen. And so the main readout, the thing we're looking for is to see if the B cells after priming are BG18 class. And so we do B cell sorting and sequencing. That sorting and sequencing was done by Shane Crotty and Devin Salk's labs. And so this is what we got. So when we looked at the germinal center B cells, we saw that in the N332 GT5 immunized animals, we found 7 of 8 of them had BG18 class B cells in the germinal centers. About 1 in 500 B cells was a BG18 class B cell. Compared to a control, this is basically a wild type trimer based on the same strain as the N332 GT5, and this did not induce any BG18 class antibodies. And when we looked at the memory B cells in the blood, we also saw 7 of 8 animals produce BG18 class B cells. So each one of these is one animal, each dot. And so you can see this red dot, non-responder, is a responder in the germinal centers, and this blue dot, non-responder, is a responder here. And so combining the data sets, it was 8 out of 8 animals that produced BG18 class responses. And because I said we defined it just by that short sequence motif in the HCDR3 loop, so we wanted to validate that if we say something is a BG18 class B cell, it actually does produce BG18 class antibodies. So Andrew Ward's lab at Scripps determined structures of 3 of these BG18 class antibodies in complex with the N332 GT5 immunogen. And so on the left here, this is the HIV broadly neutralizing antibody BG18. This is that HCDR3 loop that reaches through the sugars to contact the trimer. And you can see these 3 antibodies that we call BG18 class are nearly identical. They have the same conformation of the loop that reaches through the sugars. So we're pretty confident that when we say something is a BG18 class antibody, it really is. And in order for these antibodies to become HIV broadly neutralizing antibodies, they need to undergo a lot of affinity maturation. They need to accumulate a lot of somatic mutations. And so if we look at the somatic mutations that are happening over time, you can see across time, the BG18s are increasing their number of mutations. So this is just the percent mutation in the antibody variable gene. So we get up to about 7% mutation by week 10. And then they're also increasing in affinity. So this is a SPR plot that shows binding affinity. So lower means higher affinity. And each of these is one antibody, the affinity for the immunogen N332 GT5. And so going from week 3 to week 10, they gain about like 4 orders of magnitude in affinity. So almost 10,000-fold increase in affinity to week 10. So we think that's looking really good. And just one last thing. So I've only told you about the BG18 class antibodies that we elicited, but there are many, many other antibodies that are not BG18 class that we also elicited. And many of those we think have no chance of becoming broadly neutralizing antibodies. So we call those off-target antibodies here. So they're shown in gray. So again, this is an SPR plot, and you can see that these off-target antibodies also have very high affinity to the immunogen N332 GT5. They're comparable to the BG18s shown in pink. But we've now designed a set of boost immunogens, showing one here as an example that can selectively, they basically knock out binding to all of the off-target antibodies. So none of the off-target antibodies can bind to this boost candidate, but still more than half of the BG18 class antibodies can bind to the boost immunogen. So we think this will be a good way to selectively boost the BG18 class antibodies while deflecting the off-target antibodies. And so the conclusion, so we've now validated vaccine priming of BG18 class antibodies in non-human primates with our N332 GT5 immunogen adjuvanted with SMNP. So we think this is a good proof of principle for HCDR3 dependent germline targeting. And so the next steps are we need to now show that we can boost these BG18 class B cells to become broadly neutralizing antibodies. And so we're working on that in both non-human primates and in knock-in mice, which is what Zhenfei is going to talk about next. We're starting to work on the boosting of these antibodies now. And in addition to that, we need to know if we can actually prime BG18 class antibodies in humans. And so, as Bill had mentioned, there's a clinical trial that's happening right now. It's almost fully enrolled, called HVTN 144, where we're testing priming of N332 GT5 with SMNP, just as we did in this NHP study. We're testing that escalating dose priming strategy as well as just a standard bolus immunization strategy. And so I just want to acknowledge everybody that has worked on this. There are many people in Bill Sheaf's lab. Shane Crotty's lab has done a lot of the sorting and sequencing, and as well as Devin Salk's lab. Andrew Ward's lab has solved many cryo-EM structures for this project. And Jim Paulson and John Yates' labs at Scripps have worked on this. And then we've been working with Facundo Batista's lab for a long time on the BG18 project. And Zhenfei is going to talk about some of that work next. So I'll open it up to questions.
Thanks, John. I wanted to ask, somebody was asking about why a participant hadn't made a VRC1 class response in G001, and Chris answered that that participant didn't have, they didn't have permissive alleles for this one human gene, VH12. And I'm wondering if you want to comment on what do you think are the genetic requirements for producing a BG18 response? Like what alleles do you think are going to be crucial, if any?
So now I think it's likely just the D gene in the antibody is what I think is the only critical thing. So D33, a person would need to have D33. We used to think it also needed a specific J gene, but non-human primates lack that J gene, and they're still able to make a BG18 class response. So I kind of think that the D gene is the main thing that you have to have. Yeah.
Okay, here's another question that came in, and it's, can immunogen design be done using AI? And the person says in parentheses, dry lab and mRNA tech, or is much animal experiment and resources needed?
AI can definitely help immunogen design, but you definitely still need to test. I don't think we're to the point where we don't need to test it in an animal model to see if it's going to do what we think it's going to do. Yeah.
May I also weigh in on this?
Yeah, please go ahead, Torben.
So all these immunogens we are presenting today were kind of done before the big AI wave, so they are still doing with the old-fashioned way, if you will. But now that the next generation of immunogens and some of these boost candidates that we're working on, we are heavily relying on AI. It's kind of one of these perfect situations to use AI to design immunogens. And that's what the next generation is going to be all about. However, despite this progress, that just speeds up how fast we come up with new candidates to test, but we still need to go down the route of doing the animal experiment to find out whether they actually work as intended.
Okay, here's another question. I think this one's for John, and this is from Selasi Diletsu. And the question is, in trying to eliminate the binding to the off-target antibodies, you seem to have lost quite a bit of affinity compared to the original immunogen. How much do you think that is going to affect the quality of boosting you'll achieve? So it strikes me that that might have been directed toward Chris, but
no, I think it was
okay, perfect. Yeah, go ahead. Yeah,
it's right here. So you start with this affinity, and when you knock out the competitors, you also lose affinity to the BG18 class antibodies. But we think that's a good thing. We think the boost immunogen needs to be more native-like than the priming immunogen, and needs to have lower affinity in order to drive more somatic mutation in the right direction. So if the boost candidate still bound with this high affinity to the BG18 class antibodies, we don't think that would drive affinity maturation. You would likely just turn those cells into plasma cells that would secrete antibodies, but we don't want to do that. We want to continue the maturation. And so the epitope needs to get more native-like, which will reduce the affinity. And so that'll give the antibodies a chance to gain more mutations that will gain more affinity towards a more native-like epitope, essentially.
Okay, here's another question related to the same graph, probably. This is from Nuna Tavera, if I'm pronouncing it right. Were the off-target monoclonal antibodies unexpected, and what do they target?
They're not unexpected. So the BG18 class B cells are very rare. So we were very confident that we were not only going to get BG18. We thought for sure we're going to get an immune response to the whole trimer. But in this case, it's very surprisingly specific. We made antibodies from we clustered the off-target antibodies, and we ordered one antibody from every cluster. So they're all different genetics, but every single one of them that we tested is knocked out by the same mutations, and they are all targeting the V1 loop, the variable loop 1 of HIV. So in this particular case, it was very easy to knock them out because they were all targeting a very specific part of the trimer.
Okay, so there's another question that is I think came in after Chris and Xuan-Song, but I think they could try to answer because it will help explain the next talk as well. So the question is from Hussein Yuman, and the question is, this step of using humanized mouse model is more potent. Is this step of using humanized mouse model more potent and promising compared to unhumanized model? And how do you humanize the mouse? So, Chris, do you want to? Yeah.
Yeah.
So, normal mice don't have the correct genes that would be necessary in order for us to elicit, or in order for there to be BNAb precursors present in the mice. So we need to engineer the mice in order to have human genes that are capable of becoming BNAbs. And the way we did it, or the way Fred Alt did it, was to knock in the genes so that it's a rearranging mouse. That just gives us a little bit more diversity. And then Xuan-Song can answer how they've been doing it in Facundo Batista's lab.
Yeah, so actually our mouse models, I mentioned we isolate the precursors from human. So that is also based on the EOD-GTA, the germline targeting immunogen, as a probe to fishing those human B cell precursor VRC01 precursors. So then we knock in those precursors in our mouse models. So theoretically, the EOD-GTA germline targeting immunogen can bind to those precursors, and then we can test the heterologous, the boosters to see if it triggers the precursors and go the affinity maturations. So that I think the naive I think the wild-type mice, the non-human humanized mouse models don't have those precursors. So I think they will not it doesn't work very well for this germline targeting immunogen and the boosters.
Yeah, and it's important to note that these models are very specific. They're sort of made for purpose for specific classes of broadly neutralizing antibodies. The models that we're using most of the time are not sort of general humanized mouse models, though those can answer other kinds of questions.
Yeah, exactly. Thank you.
Okay, I think we should, there's two other questions related to mouse models, but I think we should let those come after Zhenfei does his talk. So John, thank you very much. And next up is Zhenfei Zi, who will talk about the same project, the BG18 project, and his studies using protein and RNA priming and boosting. in a different kind of engineered mouse that they made in Facundo's lab.
Can you see my slide here?
Yeah, but you should put it on presenter view. If you click the little button down at the bottom right, that one. Yep.
I can't not open my camera. Maybe the host. It's okay. It's okay.
Oh, can someone let Zhenfei show turn his camera on?
Hi everyone, I'm Zhenfei Xie, a research scientist from this lab of Reagan Institute. It's my little short story here. Today my talk is HIV-1 trimer boosters initiate a precursor to broadly neutralizing antibodies targeting Vis3 glycan. This is a cooperative project with strong support from the Shift Lab and the World Lab. As previous speakers mentioned that the germline targeting and the sequential immunization strategy designs a series of more and more native like immunogen to target the HIV BNAb precursors and drive them into BNAb like a mature antibody. The Vis3 glycan is an immune supersite. Many BNAbs are identified to target this epitope. Among them, as John Steichen mentioned, that the BJ18 is a star, which show good neutralization potency and breadth. Here we infer the BJ18 precursor antibody sequence, and we lock in that sequence into mouse models, and we generate the mouse that can express the human BJ18 precursor antibodies heavy chain. To test whether the preclinical germline targeting immunogen N332 GD5 trimer protein designed by John Steichen can activate the HIV beta precursors. Here we transferred the BJ18 naive precursors into CD45.1 congenital mice to dilute the precursor frequency. Because as John Steichen mentioned that the precursor frequency of BJ18 in human repertoire is very low. We try to mimic the human condition. Then we immunize the mice with the N332 GD5 trimer protein, and we track B cell response dynamics. After immunization, the germinal center reaction is very strong, and we could see large number of transferred BJ18 precursors get involved in germinal center for strong activation. Majority of those cells can bind to this GD5 trimer antigens. And this strong BJ18 precursor activation can last until day 49. Then we collect the serum samples and perform the NS-AMPA assay. We found both the BJ18 lineage and the wild-type lineage derived antibody seem to approach this Vis3 glycan epitope, but they are in different directions. Later, more delicate structural results showed our BJ18 lineage derived Fab contact this yellow GDIR cleft of Vis3 glycan epitope, while it's known as CDR3. However, the wild-type antibody actually binds the V1 loop, which is off the enough maturation path. That suggests the wild-type mouse model cannot generate the BJ18 class enough. The GCB cells are undergoing selection, somatic hypermutation for affinity maturation. To track the BJ18 precursor maturation process, we sorted those CD45.2 antigen binders and perform BCR sequencing. And we found the BJ18 heavy chain gain a lot of somatic hypermutations. And those mutations are highly enriched in the CDR. Now we have proved that the N332 GD5 trimer protein can efficiently activate the BJ18 nice precursors in low precursor frequency. And we know priming BNAb precursor is just the first step. Next, we want to see whether the priming immunogen GD5 can pass the baton of activated BJ18 precursors to the booster immunogen for further activation. So here we transfer the PG18 knife precursors. We prime the mice with the GD5 trimer protein. Later we boost them with B11 trimer proteins. After protein boosting, the GC size increases dramatically, and we could see relatively more expanded PG18 precursors in the germinal centers. Majority of them can bind to this B11 antigen probe. But just like around half of those samples can have a good response. The B11 protein booster can further increase the PG18 heavy chain amino acid mutations. More mutations are enriched in the CDRs. Moreover, and we found after B11 boosting, we found some PG18 lineage-derived Fab contact the B11 trimer protein in a similar mode as mature PG18 Fab by the native-like protein BG505, suggesting boosting with the B11 trimer protein can further increase PG18 precursor B cell somatic hypermutation and affinity maturation. The SARS-CoV-2 MR vaccine exhibited excellent performance during pandemic, providing a new paradigm for next generation vaccine development. To test whether our GT5 immunogen in mRNA formulation can also trigger good BNAP precursor activation, here we transfer the PG18 knife precursors, then we prime with the membrane anchored N332 GT5 mRNA immunogen. After GT5 MR prime, we could see robust PG18 precursor activation in the germinal centers, and this strong activation can last for a very, very long time. Now we have evidence to show that the GT5 MR immunogen can mirror the immunogenicity of GT5 trimer protein to efficiently activate the PG18 knife precursors. Then we wondered, what if we deliver both the prime and booster immunogen in mRNA MP formulation, where we still get a good or even better response? Here we transferred PG18 knife precursors. We prime mice with GT5 MR immunogen. Later we boosted them with B11 MR immunogens. After MR boosting, the GC size increased rapidly, and we could see more expanded PG18 precursors in the germinal centers for activation. Majority of those cells can bind these B11 antigen probes. Most of the samples have a good PG18 precursor activations on day 58 after B11 boosting, and this strong BNAP precursor activation can last at least 36 days after B11 MR boosting. To summarize this study, here we evaluate the efficacy of preclinical germinal targeting immunogen N332 GT5 trimer protein, and we found this protein immunogen can efficiently activate PG18 knife precursors in low precursor frequency. We also tested this immunogen in mRNA formulation, and we found this mRNA immunogen can largely recapitulate the activation and the maturation process of PG18 precursors as with the GT5 trimer protein. Moreover, we performed the first boosting study with the B11 trimer protein, and we found this protein booster can further fuel PG18 precursors in the germinal centers for activation. Lastly, we found if both the prime and the booster immunogen were delivered in mRNA formulation, this regimen can work at least as efficiently as the protein immunization regimen, if not better. After prime and the first boost, we could see the PG18 lineage heavy chains gain somatic mutation over time, and they are on the right track for affinity maturation. Some of them began to show neutralization activity to intermediate native-like pseudovirus, but not fully wild-type virus yet. The artist also helped us with the graphic design for the take-home message. Here, the dragon body composes of the RNA loops representing the mRNA immunogen. The phoenix body is formed by the protein trimer, standing for the protein immunogens. The flames firing the pearl symbolize that those prime and first booster immunogen can instruct and shepherd the PG18 knife precursors. I thank participant lab member especially my mental faculty for the strong support and Dr. Yunqi for the invaluable contribution to this study. I thank all the Scripps collaborators, particularly Professors Bill and Andrew, Dr. Joe Steichen, and Gabe Odorowski for the amazing cooperations. I thank Moderna for providing us with mRNA immunogens, and I thank all the funding resource for supporting our studies. And that's all from my part, and I'm happy to take questions. Thank you.
All right, thanks, Zhenfei. So there's a question that I think you should try to answer, and then maybe John can try to answer also. And the question is, how comparable are mouse and primate models? To put the two presentations in perspective, why are you going back to the humanized mouse model? I think that was, in relation to John talked about NHPs, and now you're talking about mice. So do you want to comment on that?
I think for the humanized mouse model, we can, put exactly the same precursors into mouse to evaluate the immunogen trigger the BNAP precursor response. And for the monkey stuff, I think John maybe can comment that. Yeah.
Yeah, sure. So for the knock-in mice, I would say it's pretty accurately predicts priming in probably one boost. And so you can do sort of iterations where you can, after you do one boost, then you can make a new knock-in mouse to go iteratively. Whereas in NHPs, I think you can work out the full sequential regimen, but obviously the NHP studies are much more expensive and they're more time consuming. And so we like the NHPs a lot because we just think they're accurately reflecting what you're going to get in humans. But the mouse studies are just so much faster and cheaper, so they definitely have an advantage still in many ways.
Yeah, maybe I could add that I think using the knock-in mice, if you know the relevant human genes, which we always do, then Facundo and Fred Alt and others can always make a mouse model that's relevant. But for non-human primates like rhesus macaques, in some cases, they don't have the capacity to make precursors of a particular class. And so the one example would be VRC01 class BNAbs, rhesus macaques. If they make them, they're exceedingly rare. And so the immunogen that we developed that did work extremely well in humans doesn't produce any VRC01 class responses in rhesus macaques. So there are examples where mice are the only tool available, at least macaques don't seem to be helpful. And I think there are other BNAb classes where the genetics in the macaque is somewhat similar to humans, but not quite. And in some ways, doing things in a macaque could be more difficult, like the way John said, could be more difficult than in humans, in which case, if you're successful in macaques, you can feel pretty good about your chances in humans. But there are other cases where the genetics of the macaques makes it a bit easier. They have genes that make induction of BNAbs even easier. And I think that's true for the apex class of BNAbs. And in that situation, even if you are somewhat successful in macaques, it's not so clear what that means for what's going to happen in a human. So, I think the major advantage of the engineered mice is you can try to make them very specific to have exactly the human genes that are relevant to human vaccination. But obviously they're very small and they're not used to long vaccination regimens, like John, that the point John was making is it's not clear to us that you can study very long, elongated multi-vaccination regimens in mice. Okay, so there's another question. Yes, go ahead, Torben.
Could you also tell us a little more about, if we already had the results from protein, why did we do it again with mRNA LNP?
In this in our mouse model, I mean at the primary stage we see both the protein and mRNA works very well with this mouse model, but we find some some kind of some difference after first boost with between the protein and mRNA immunization. After if in this mouse model, if we give a booster delivered in mRNA, we can find the response is more stronger than protein I see in this in these mouse models. And also because as I mentioned, because the mRNA combination immunogen could be well established, could be easily to adapted and to produce the antigen, and also it's kind of be easier to produce in large scale. I think it's maybe more useful in practice for future usage in humans. Yeah.
Okay, Torben, was that an acceptable answer?
Sure.
Okay. Okay, here's another question that came from William Koazaza. And he asks, is it possible that boosting immunity might stimulate the body to produce an autoimmune response?
I think for HIV BNAbs, because a lot of HIV BNAbs have a very long HCDR3, and also the mature BNAbs kind of have some reactivity. Some studies show that the BNAbs of HIV some kind of auto immunity effect. That could be possible, but we still can generate a lot of good BNAbs that to avoid the auto immunity side effect. Yeah, we have to evaluate that kind of side effect in the future. The human or monkey clinical in future human clinical trial. Yeah.
Yeah, I think I agree with what Zhenfei said. I think when we're immunizing with these immunogens, we're not using immunogens that are known to be autoantigens, even though there is some argument in the literature that BNAb induction faces difficulties of being blocked by autoimmunity. That is, we haven't seen evidence of that in our studies. Let's say that, but I agree with Zhenfei that that's obviously why we're doing phase 1 clinical trials, and we'll see if anything comes like that. Okay, I think we should move on. There's one other question, but I think we can leave it for the end. So let's move on to the last talk, which is from Torben Schiffner.
Okay, so I'm gonna show you a different epitope that the others haven't covered yet. That's called the EMPER, the membrane proximal external regions. And this epitope, it's got some advantages, but it's also got its real own challenges that are quite different from the other talks we've heard today. So the EMPER is the membrane proximal external region. And this is the envelope trimer here in blue. And you have the membrane here at the bottom, and the EMPER is shown here in purple, the membrane proximal external region, so directly next to the membrane. And because it's so close to the membrane, the whole envelope trimer is kind of in the way. So for the antibody 28 shown in gray here, to get access, the whole thing kind of has to lean over a little bit to get access, so the antibody can access this region. But on the other hand, The emperor is highly conserved, so antibodies that target this region, such as the one called 10E8, they are very broad. They can neutralize like 98% of the viruses out there. But we have to deal with this problem of the epitope not being well accessible. And 10E8 solves this issue by kind of having a long protruding HCDR3 loop, similar to what John showed us earlier. It's very long and it kind of grabs into this this recessed area here to hold on to this conserved epitope. But the mature broadly neutralizing antibody, it's gone through many rounds of affinity maturation. It's really learned to recognize this occluded epitope. But if we want to do germline targeting, the precursors won't have learned this ability yet. So we kind of need to make it a little easier for the naive antibodies to recognize this epitope if we want to consistently induce epitopes against this region. So our approach is called germline targeting. We're using epitope scaffolding here. What that means is, because it's so difficult to access this region, we cannot take it out of its normal environment and just present this epitope on its own. However, to do that, we kind of have to come up with a way to stabilize this little bit of protein, this little piece of HIV. We need to stabilize it in this conformation that it has when it's bound to the antibody. And we do this by this epitope scaffolding, where we build a epitope around it. Let me show this again here. So we've got 10E8 in green now, and we had a structure of its epitope, this little blue region here. And to stabilize this epitope in this region, we kind of build a little scaffold around this epitope to stabilize it in the bound conformation. And that's quite different to what the other people showed, because their epitopes are much more accessible on the envelope trimer. But similar to what the other speakers have shown us before, if you do this, if you take the little epitope out of its normal context, you graft it onto this epitope scaffold, you stabilize it, and you test binding, the mature 10E8 antibody now binds really, really well to this epitope scaffold. And again, good binding is at the bottom, no binding is at the top. However, the precursors of these antibodies that haven't gone through affinity maturation yet, they don't bind at all. And that's so epitope scaffolding on its own is not going to induce these types of antibodies. But now we combine the epitope scaffolding with the germline targeting that the others have talked about. And we spent many years optimizing this immunogen, and we came up with, took us 10 iterations, our germline targeting version number 10, the 10th generation of these immunogens. Now, this one binds to many precursors. It also still binds to the mature antibody 10E8, but the binding got a lot weaker in the process. So again, we spent another year kind of going backwards and then optimizing again. And after many, many iterations, we ended up with GT12, which binds to about half the precursors that we tested it against. And it also binds really well to the mature broadly neutralizing antibody. So now this is an immunogen that we think it would activate these precursor B cells and kind of activate them and drive them towards, start the process of maturing towards the broadly neutralizing antibody, the mature antibody. So now that we have this immunogen that we made with a combination of computational design and in vitro selection, now that we have it available, how do we test it? Well, we need to see whether it induces these 10E8 class antibodies. And just to reiterate, 10E8 class means we have this long HCDR3 loop shown in gray here with a specific binding motif at the tip of this long loop. So that's what we're looking for to induce. But similar to the other speakers, there's not really a perfect animal model out there. So really, we'd like to test it in humans, but you can't just put a new immunogen into humans. You first want to run some tests. And since we can't put the immunogen into humans, we just took B cells out of humans. We just take the blood, isolate the B cells from the human blood, and then we test which of these B cells bind to our immunogen. And so we sort out the ones that bind our immunogen. This was a collaboration with the Quadi lab. And then we check these antibodies that they pulled out. Do they have the properties we're looking for? Are they 10E8-like? And the first thing I said, we need this 10E8 binding motif in the HCDR3. It's called YXFW. I'm not going into the details, but how many of the B cells have the binding motif? Generally speaking, we'd expect about 2% of all B cells to have this binding motif. But among the B cells that bind our immunogen, we are looking at over 80% of all B cells, so massive enrichment. The B cells that our immunogen pulls out of human blood, most of them, almost all of them, have this 10E8 binding motif. So it really does that really well. The other thing is, I said, we need a long HCR3 loop, but the motif alone is not good enough. It also needs to kind of grab into this recessed epitope with a long HCR3. So the normal length of this loop is around 14 to 15 amino acids, and this is like what a normal length distribution looks like. And we are looking for a length of like 21 to 24 amino acids shown in gray here. So this is the length that we'd like to get. And as you can see, normally among B cells, it's really rare to find B cells with this length. But now what about the B cells that bind our immunogen? Well, they had a length distribution shown here. So really focusing on these long HCDR3s. So we're pulling out long HCDR3s with our binding motif, exactly what we were targeting. So this looks really good, but now we want to test what if we immunize with this immunogen. And we made this small protein, and the immune system kind of doesn't quite take it very seriously if we just immunize with this really small protein. It doesn't look quite like a virus. So to make it more virus-like looking, if you will, we fused it to a self-assembling nanoparticle. So we just took our immunogen short linker, a self-assembling nanoparticle, another linker, and another immunogen. So we have the nanoparticle core in the middle and two of our immunogens. And now this part in the middle assembles into a 12-mer. And what you get in the end is this nanoparticle here that has 24 copies of our immunogen. And now it's kind of bigger, it's multivalent, it looks more like a virus. So the immune system will make a much stronger immune response against this. So with this in hand, we went ahead and immunized animals. And we started off with mice, and we immunized these mice. And again, we looked at how long are the HCDR3s we're getting out. The control immunogens here, we're getting these 14, 15 amino acid long HCDR3s. But with our immunogens, we get these long HCDR3s. And in total, in these knock-in mouse models, if you immunize with control immunogens, you never get 10E8-like HCDR3s. But if you immunize with GT10 or GT12, about 5% of all B cells that are triggered are the ones we're looking for. And this works as both protein or mRNA. And the mRNA, as mentioned before, has a much easier path to the clinic. It's much easier to put this into humans with mRNA. We also tried it out in monkeys, in rhesus macaques, and we got a very similar result here, where both GT10 and GT12 induced 10E8 class responses in non-human primates. And this is a very tricky animal model because we optimized this immunogen for human genes. The monkey genes are a little bit different, and so it's harder here. They are not as frequent as in humans. And despite it being much more challenging than in humans, we believe, we got a robust induction of 10E8 class responses in monkeys. But we even got a little bonus at the end. There's a second type of antibody called LNO1, shown in green here, that also recognizes the emperor in a slightly different conformation, but it also has a very similar binding motif that is shown here at the top of a long HCDR3. So again, we have a long HCDR3, we have a related, not identical, but related binding motif, and it binds to the same place of the envelope protein. So we looked at maybe we didn't design for it, but maybe we also get these LNO1 class antibodies. So we went back to our old data set and looked at, do we get LNO1 class responses in humans if we saw human B cells? And the answer was a resounding yes. If we these B cells are supposed to be very rare, one in like 5,000. We get them, about 5% of the B cells we pulled out are LNO1-like. And again, we looked at the same immunization we already did in non-human primates, in monkeys, and we trigger these LNO1 class responses in most of the monkeys we looked at. So we kind of got a freebie here, 2 for 1. We not only get 10E8 class, we also got a second class of broadly neutralizing antibody called LNO1 class that's related, though somewhat distinct. To summarize all this, I've shown you that we can make these epitope scaffolds that kind of take the epitope out of its normal context to better present it to the immune system. And we can engineer them using germline targeting as a strategy to specifically engage antibodies with the predefined features. I've shown you that we can use them to isolate 10E8-like and LNO1-like antibodies from human blood. And we can prime 10E8 class and LNO1 class precursors in both non-human primates and in stringent mouse models. And finally, I've shown you that we can deliver them both as protein and the much more translatable mRNA LNPs. And therefore, we think that these merit clinical evaluation as a germline targeting HIV vaccine prime. And with that, I'd like to thank everybody involved. Bill, Kimo, and Olivia from the Chief Lab, collaboration with many other labs, the Batista Lab, the Quali Lab, the SOC Lab. And of course, I'd also like to thank all the funders without whom none of this would have been possible. And with that, I'm happy to take any questions.
Thanks, Torben. Okay, here's a question. Scaffolding makes the epitope more accessible, allowing for the production and identification of relevant NAbs, but in vivo, the epitope is still hidden. I think they mean in vivo on HIV. The epitope is still hidden and NAbs rare. Does this suggest the approach is more appropriate for passive immunization?
So it's absolutely true. It makes it much more accessible. So what we need to do here is, I've only talked about the priming immunogen here. We need to now boost with immunogens that get progressively more native-like. So the priming works really well, and we make it much easier for the immune system to recognize this system, this epitope. However, we're now working on the boosting immunogens, and at some point we need to bring in all the other requirements like the membrane and the envelope trimer to make it as difficult to access as it would be in vivo. And so we are going for the stepwise approach where we first teach the immune system to recognize this epitope, get really high affinity for this particular spot, and then bring in the other parts that make it more difficult. And that's how we are planning to overcome this additional difficulty of the recessed epitope. So it is true that for passive immunization, it's much easier because you don't have to teach the immune system everything. You just give the antibody. But here, with the stepwise approach, we are hoping to teach the immune system to first recognize it, first make it easy, and then come in with the more difficult surroundings. I'd like to kind of compare this to, if it's really hard, it's like rocket science. And if you want to teach a kid rocket science, then you don't just start off with the university course on rocket science. No, you first teach them basic mathematics, and then you go to middle school math and high school math, and then you start the university course. So if you want to do this really difficult thing, you start off easy and you make it progressively harder.
All right, thank you for that. And I think just to add to to the person who answered the question, to ask the question, we do know that the 10E8 Emperor BNAb can protect against infection in a non-human primate model. And so what Torben's trying to do is induce antibodies like that, so that if he's induced them, then if the person is exposed to HIV, the BNAbs in their blood would protect against actually being infected. That's the idea. And that sort of answers another question that came up recently. So there is another question that's a kind of a tricky one. And so I'm going to try, I'll give the question and I'll give some answer, and we'll see if any of the other speakers want to take a shot at it. This is from Pedro Garbez. And he says, what is your expectation for such vaccine candidates to alter the clinical evolution of HIV infection if they are not 100% efficacious? And then he goes on to a more tricky question that says, in considering current HIV therapeutic guidelines, how would you approach a study to assess such impact in those patients who got infected by HIV in spite of the HIV prophylactic vaccination? Thanks. So if I could start, I would say we're under no illusion that the vaccine will be 100% efficacious. No, I don't think any vaccine is, or very few. But we do aim, we're hoping for 70 or 80% efficacy. And to me, what that would mean is everyone who's vaccinated, they would they'd be protected, 70 or 80% of the time if they're exposed to HIV, if you will. So it should reduce transmission overall. Depending on the fraction of people at risk, how many of them are actually vaccinated, that'll be one parameter in how that affects the overall clinical evolution of HIV. The flip side of the coin is for individuals that are vaccinated and they have some levels of broadly neutralizing antibodies, maybe against multiple epitopes, but let's say the levels are not high enough to prevent infection. They get infected, and then the virus is evolving in the presence of sort of low or medium levels of BNAbs. You can imagine that many different things could happen, and I don't think we really know the answer. One thing that could happen is, the virus could mutate away from those BNAbs and become more resistant to BNAbs. That could happen. But if we are inducing multiple different classes of BNAbs, the virus is going to take a pretty substantial, we know that it can mutate away from any one class, but I think it's going to be tricky to mutate against 3 or 4 classes. So it might take a big fitness hit. And so, in fact, even at modest levels of BNAbs that don't prevent infection, you could imagine it's possible it could sort of dampen the impact of infection. It could slow the progress of the infection. It could allow other arms of the immune system, like T cells, to find infected cells and kill them. And I think what could happen, the dynamics of what could happen are very complicated. And I don't really want to, it's not my expertise. And I think it's also very complicated. So I think it'd be very hard to map out studies to do that. But probably there are other people that could take that up. That's not really our job. I don't know if anybody else wants to take a stab.
May I just wanted to add that any vaccine that at 70, 80% efficacy would make a big impact on the transmission rates. And we're not suggesting the vaccine would be like a standalone that solves it all, but we are still going to keep treating people infected with ART and so on. So it would be a fantastic additional tool in our armory.
Yeah, on top, and of course, on top of long-acting PrEP, which will, is going to become probably standard of care at some point, assuming, it gets rolled out and is accessible, which we're obviously totally supportive of. Okay, there's another question that says, is there a thought about eliminating HIV receptors to prevent HIV replication in the human body and prevent infection. You want to try that, Torben?
Well, first of all, it's not our area of expertise. I think like this would be some kind of genetic modification of the host, and doing this, the HIV vaccine is targeting people who are not infected yet. Doing something this such a big change to the genetics would be quite risky in people who are not infected, in healthy people. So I think this is more something that people working on a cure are looking into to get people who are infected to rid them of the disease rather than doing it to uninfected people.
And a few people have received transplants that from donors that lack the CCR5 co-receptor, bone marrow transplants because they were ill, for example, with leukemia otherwise. And that does make them immune to HIV infection, but that's a very complicated and that's a risky treatment. The risk of that procedure alone is quite high. So to support what Torben was saying, doing that kind of thing on healthy people is not regarded really as a global health or even as a generally applicable prophylactic intervention. Okay, here's one more question. It is clear that preclinical research is moving fast at a pace that clinical research will struggle to cope with, in parentheses, manufacturing and trial length. What could we do to ensure that we are testing the best products in clinical trials and not the one before or even the one before that? You want to try, Torben, since your face is on the screen?
Sure. I mean, we are constantly improving these things. I think we can do a lot of these studies preclinically, and so we gain valuable feedback from each of the clinical studies we do, no matter whether it is the latest or the one before that. And we are still working towards the point where we have the entire regimen from prime to all the boosts mapped out preclinical. And once we have that set, then we are going to take that as one regimen and put it into the clinic in one go. I think that that's going to be a watershed moment once we're ready to do that. But even with, if it's not the final version, we learn so much with each human clinical trial we do.
Yeah, and I would say our sort of philosophy is to really put the screws on the problem in the preclinical space and to try to mimic key parameters of human vaccination as much as we possibly can and develop vaccine immunogens that pass very stringent tests, just like the test that Chris and Xuansong and John and Zhenfei and Torben have showed you. We go through, with a lot of collaborators, we go through extremely long lengths to make sure we believe that if the immunogen is successful in these animal models, they do have a quite good chance of performing the way we want in humans. And what I would say is stay tuned for the results of our human clinical phase 1 trials. G001 was definitely successful. And people should stay tuned to see what happened with G002 and G003, and also HVTN 144 and HVTN 302. We're trying to make it that when we go to humans, we keep learning, but we also confirm what we thought was going to work so that we can move from basically one success to another. And so to the person who asked that question, I think we are trying to take the best thing to the clinic, at least what we regard as the best thing. And so maybe that's probably a good way to close out and hand it back to Asli and I just want to thank her for organizing, a really great session.
Thank you so much. On behalf of the Global HIV Vaccine Enterprise, I would like to thank, of course, the audience for your interest and for your engagement, the presenters for really comprehensive explanations, and thank you so much, Bill, for helping us put this together and for your brilliant moderation. As I said, the session will be available on IIS Plus soon. So look out for it, and we hope to see you soon for future webinars. And with that, please have a very lovely evening, day, whatever it is.